ये वीडियो क्रोमोसोल म्यूट से संबंधित है जिसमें डिलिटेशन, इन्वर्शन, ट्रांसलोकेशन को समझाया गया।
The human body is built upon a microscopic library of information. Zoom into the nucleus of a single cell, past the billions of base pairs, to find the genome's physical foundation, the DNA double helix. This molecule is physically volatile. During every cycle of replication and division, double-strand breaks and recombination errors physically alter the organization of the chromosome. Geneticists categorize these large-scale rearrangements into four principal types: deletions, where code is lost; duplications, where it is added; inversions, where it is flipped; and translocations, where segments trade between non-homologous pairs. These structural variations are responsible for a wide spectrum of clinical pathologies. Mastering their meiotic behavior and phenotypic consequences is a primary requirement for the CSIR NEET inheritance biology syllabus. Chromosomal deletions involve the absolute loss of a genomic segment. A single break at the end of a chromosome arm causes a terminal deletion, while two breaks along the same arm can excise an internal segment, resulting in an interstitial deletion. When a segment is lost, the cell is left with a single functional allele. For many structural proteins and transcription factors, this 50% reduction in protein concentration is insufficient for normal development. This is the mechanism behind Cre-Du-Chat syndrome, where a deletion on chromosome 5p disrupts laryngeal and neurological growth. Deletions also facilitate pseudodominance. By physically removing a dominant wild-type allele, the deletion allows a recessive mutation on the homologous chromosome to be expressed in a hemizygous state. This serves as a vital diagnostic tool in the lab. In Drosophila, if X-ray irradiation of a red-eyed male leads to white-eyed offspring, when crossed with a recessive female, it suggests the radiation deleted the dominant eye color locus on the X chromosome. The impact of a deletion is determined by two factors: the essential protein levels that fall below a physiological threshold and the recessive traits that are no longer suppressed. Duplications often arise from non-allelic homologous recombination. During meiosis, highly similar repetitive sequences can cause chromosomes to misalign. When they cross over, one chromosome loses a segment, while the other receives an extra copy. Having 150 in Charcot-Marie-Tooth disease type 1A, a tandem duplication on chromosome 17 leads to the overexpression of peripheral myelin protein 22, resulting in the progressive demyelination of nerves. Cellular health depends on strict quantitative balance. Whether code is missing or duplicated, the resulting dosage imbalance leads to systemic dysfunction. An inversion occurs when a chromosome segment breaks, rotates 180 degrees, and is reintegrated. Because these are typically balanced rearrangements with no net loss of DNA, the carriers usually show no physical symptoms. The difficulty begins during meiosis. To achieve gene-for-gene alignment with a normal homologue, the inverted chromosome must contort into a characteristic inversion loop. Inversions are often called crossover suppressors, but recombination still occurs inside the loop. However, a crossover within a paracentric inversion loop produces a dicentric bridge and an acentric fragment. These structures are physically unstable and lack a balanced set of genes. Because the resulting recombinant gametes carry lethal deletions and duplications, they never produce surviving offspring. This makes the inversion a functional lockbox for linked genes. A translocation involves the exchange of segments between non-homologous chromosomes. In a reciprocal translocation, two different chromosomes swap pieces, usually resulting in a balanced genome for the carrier. During meiotic prophase, the swapped segments force the four chromosomes into a cross-shaped quadrivalent. Only alternate segregation produces viable offspring. Here, centromeres pull diagonally, ensuring a complete genetic set. Any other pattern leads to unbalanced gametes and semi-sterility. Robertsonian translocations are a specialized form involving atlas antrochromosomes, such as 14 and 21. The long arms fuse into one large chromosome, while the redundant short arms are lost. A carrier of a Robertson translocation has exactly 45 chromosomes, but remains healthy because no critical euchromatin was lost. However, they carry a significant risk of producing gametes with an extra copy of chromosome 21, leading to familial Down syndrome. These rearrangements allow a balanced genome to persist in a carrier, only revealing the structural imbalance when chromosomes are partitioned during reproduction. Beyond these singular breaks, we now observe catastrophic genomic reorganizations. In chromothripsis, a single chromosome shatters into dozens of fragments during a single-cell cycle and is randomly stitched back together. These failures are often localized at specific genomic coordinates. Regulatory proteins like PRDM9 guide the location of meiotic breaks, meaning the susceptibility to these rearrangements is itself encoded in the genome. Deletions, duplications, inversions, and translocations represent the fundamental ways the physical structure of DNA can fail. The study of these mutations provides more than just clinical insights. It defines the mechanical consequences of chromosomal instability within the human cell.